Hi scientists, it's Miss Fulton again. Today I'm going to walk you through codominance and incomplete dominance. So I'm going to start by doing page 22 in your workbook, and then I'm going to do some notes on lined paper. So you should grab your workbook, and then if you want to write things down, grab a lined piece of paper. But if you just want to listen, I will post a picture of my notes on our Teams in the genetics channel after this video. Okay. So far we have been learning about something called complete dominance. This is also called Mendelian traits or dominant recessive traits. But this is where you have one allele that's dominant to the other. So in this case, red with a capital R is completely dominant to a white flower, which has the allele is little r. So if you have the heterozygote, you're going to have big R, little r. And so far, the examples we've been doing means that this big R wins because it's completely dominant. So our phenotype would be red. So this column, the middle column on this page, these are all the heterozygotes. And this is where the difference is between these three types of dominance that we're going to learn about. Okay, so heterozygotes so far have been whatever the dominant phenotype is. However, that's not the case in every type of, not every situation. So you can also have incomplete dominance. Sometimes this is written as a big capital letter with a little, like almost an exponent. You can also again, just write big R for this one, as long as you know what kind of dominance it is. So in incomplete dominance, what happens is you get your dominant, you don't have a dominant allele, you have just two different alleles. So in this case, we have red, we have white. You could write WW if you want, totally up to you. But if you do this, you should have all capital letters because one isn't dominant over the other one. This lowercase letter is to represent that it's recessive, but there is no recessive allele here. They're incompletely dominant. And what happens is your genotype, I'll write it the same way they wrote it for clarity. When you have one red allele and one white allele, you get an intermediate phenotype, meaning something in the middle of the two other phenotypes. So in between red and white would be pink. So in incomplete dominance, you have a mix of the two other alleles. Okay, so you have a red allele, a white. The last type of dominance we have here is called codominance. So again, you don't have a recessive allele, so you're not using a capital and a lowercase. You can use a letter with a exponent, or you can just use two capital R's, two capital W's, it's up to you. But what happens with the heterozygote here is that both of the alleles are expressed. So when you have a red flower and a white flower, you're gonna end up with a phenotype that's half red, half white. So your genotype here will be one red allele, one white allele, and then the phenotype is going to be half red, half white. Or what might be more common is if you have petals on your flower, some of them are red or blue in this case, and some of them are white. So it might look something like that, alternating. Okay, so I'm going to work you through some examples of incomplete and codominance. Please ignore my scribbles. I started making this video and realized I had made a mistake. So in dogs, a white parent and a black parent produce a gray puppy. What are the genotype-phenotype ratios for the offspring of a white parent and a gray parent? So the first thing you have to do is decide what kind of dominance this is. So we have white and black, and they produce gray. And you have to decide, is gray an intermediate phenotype between black and white? Or is it one where both of the alleles are expressed? And gray is between black and white. So this is incomplete dominance. So what that means is that we can pick a letter. Let's do W for white. So a white parent is WW. A black parent, let's choose B. So it'd be BB. Remember, they're all capital letters because one isn't dominant over the other one. And then when we have gray, it's going to be one of each, one white, one black. Now you choose which letter comes first, W or B. It doesn't matter, just be consistent. Don't put BW for some and WB for some because you're going to get confused. Okay, so we're going to use a Punnett square again. Remember to make your hashtag sign. There we go. So let's put my white parent on the top and my gray parent on the side. And now we'll just cross them. So WW for those two on the top. And then I said that gray is WB, so I'm going to not put BW, I'm going to put WB. Okay, so for my genotype ratio, I am going to say that 50%, half of my squares, or 2 out of 4 squares, are WW. The other 50% or half are WB. And then for phenotypes, WW is white, so 50% are white, and 50% are gray. Okay, so let's try the second one. So black chickens and white chickens have chicks with a mix of black and white feathers, and these kinds of chickens are called speckled. What are the genotype and phenotype ratios of 2 speckled chickens? Okay, so let's see, we have black, white, and speckled is some black feathers, some white feathers. So let's go back to our chart and decide, is some black feathers and some white feathers incomplete or codominance? Now hopefully you're thinking codominance, that's true. So codominance. So I'm going to pick some letters again. I'm going to use only capital letters. I can use B and W again. That makes it less confusing. Black chickens, white chickens, speckled chickens, we'll do WB for speckled. Okay, so what are the genotype phenotype ratios of 2 speckled chickens? So make your Punnett square. Sorry that my hand makes it in shadow. Okay, so 2 speckled chickens. So we'll have 1 speckled chicken on the top and 1 on the side. Oh, this is one of the fun ones. So what we end up here is just 2 W's touching 2 W's. This square is touching a B and a W, but I'm going to put it the same order, WB. This one is also WB, so this is why I'm saying pick one that goes first, because if you wrote this one as WB and this one is BW, they would look different, but actually they're the same. And then this one's BB. So my genotype ratio is going to be, I have 1 out of 4 squares WW, so that's going to be 25% WW. Okay, I have 2 out of 4 squares, or 50% WB, and I have 25% that is BB. So my phenotype ratio is going to be the same percentages, 25% white, WW is white, 50% WB means 50% speckled, and 25% BB means 25% black. So it's going to be a colorful group of chickens. Okay, I hope that helps. Feel free to go on to the next item on your to-do list and let me know if you have any questions. Thank you.